Thanks to the organizer for giving me a chance to talk a little bit about the work we start doing on using nanopore sequencing. As most of you are familiar, genetic disorders can be caused by a variety of types of mutations. And in our diagnostic lab, we typically use exome sequencing. And I think our diagnostic yield overall is maybe 50%. So for half of the patients, we typically do not find a mutation. And the reason for that is that there's various different types of mutations. There can be variants that affect splicing. We might miss them in an exome, or they could be very hard to predict what their effect of splicing is. There could be complex structural variants. There can be variants that affect enhancers, non-coding regions. So typically you miss those using an exome. And if you would use short-read whole genome sequencing, you might pick them up, but you might not be able to interpret what their effect is. So one of the things we chose to do a little while ago is that that RNA sequencing might be a nice complementary test at this moment to study what the effects of variants are, even if you don't pick up the variants. So the way we do this is we use a method called Outlier, developed by the lab of Julian Gagneur in Germany, and we modified this. So what we're using fibroblasts of undiagnosed patients, and then we perform a z-score analysis where we compare the expression of genes, but also exons and introns to a reference cohort. And the reason we're also using these z-scores, I'm not using my pointer, there's no point, I guess, but if we're using the z-scores for exons, we use that to pick up sub-gene changes in expression. And we use intronic z-scores because they would go up if you would see increased intronic in retention, for example, or pseudo-exons. So that worked quite nicely. So we've, in this study reported here, Decker et al., we did this for about 70 cases of neurodevelopmental disorders. And we take a skin biopsy, we grow fibroblasts with and without cyclohexamide to look at nonsense-mediated mRNA decay. And then we performed bioinformatics analysis. And then we have a web application that our bioinformaticists developed. So this is sort of an idiot-proof way of analyzing the data. So even people that are untrained in bioinformatics, like myself, can analyze the data. And then we get nice plots like this for a patient. And we can look at quality parameters. We can select the panel that we're interested in, for example, an intellectual disability panel. And then we can look at these volcano plots. And these are very helpful to show which genes are most significantly downregulated or which exons are most significantly downregulated. And then once we pick a gene like that, we can look at other plots to see what is going on there. For example, if all the exons are down or only one exon is downregulated. But to give you one example of how this would look like in a patient, so this is a control sample where we did not find using exome analysis a causative mutation. And then we did RNA sequencing and we find that this MFSG8 gene, there's 2 exons that are very strongly downregulated. And if you look at the lower left at the sashimi plots in the top 2, the red and the blue sashimi plots, you see that 2 exons are missing. One is completely missing and the other is mostly skipped. And then the question is like, why this is happening in this patient? And it turns out that there is a homozygous variant in the exome that was classified as likely benign because it doesn't have any splice prediction whatsoever, but it was a quite rare variant, it was homozygous. So we next tested this with an exon trapping assay, and we find actually that this causes a skipping of that exon. And these are typically the variants that are very hard to predict. So our diagnostic routine flow does not have a good way to catch those variants. So we pick them up with RNA-seq. So we've been doing this. Our overall additional diagnostic yield using short-read RNA sequencing is about somewhere between 15 and 20% extra. So in this case, this was a splice enhancer. So those are, I think most of you are probably familiar. Splice enhancers are particularly difficult to predict. They're easy. I guess if you use a program like Alamut, you will see a lot of changes for many different variants, but which ones are really bona fide variants that have an effect, that's hard to figure out. So looking in hindsight, if we know the variant has an effect and we go back to the prediction, then the prediction tells you there's an effect, but it would also tell you for 99 other variants that don't have an effect. So, okay, so now back to the reason I'm here, I guess, is we are exploring nanopore sequencing for a couple of research applications, including detecting repeats, looking at methylation episignatures, for example, for Kabuki syndrome, complex variants, and particularly also for RNA, obviously. So currently the DNA we're using for this is the same DNA we use for our diagnostic flow. And we use control samples for this. And I guess this is the pipeline that we're using. I won't talk about this for long, and this is the typical data quality that we're getting. So we're quite happy with this. And now, so these are our research applications that we're testing. And since we have the machine anyway, we're also starting to use this to look at RNA sequencing long reads, and also for cases where we use short read RNA sequencing, and we find a clue of what's going on, what the mechanism is in the patient, but we cannot find the variant. So I'll give you some examples of that. So this is a case of Treacher Collins syndrome. It's a very well understood genetic disorder that's caused typically by variants in a gene called TCOV1. So we have a family here with 2 unexplained cases of Treacher Collins syndrome, and no causative variance was found using exome analysis. Even with short read whole genome sequencing, nothing was found. So we performed RNA sequencing, and this is what the analysis looked like. We see that the TCOV1 is the most, 3 exons of TCOV1 are the most downregulated exons of all transcripts that we detect. So there must be something going on with the TCOV gene. So then we went to, if you look here in the sashimi plot, what you see is that there's an alternative 3' UTR that's expressed in the patient. Control sample, I should say. And then if we, so wait, spoiler. So what you see here is that there's a weird transcript there in half of the reads. So they jump into an intron to a weird exon. That's actually not an exon. It's actually probably some kind of ALU-like element. And then if we then take the DNA and we perform nanopore sequencing, what we see in single reads is that there's a 3.5 kb insertion, an SVA actually, that's probably the cause in this patient. And we confirmed this by diagnostic DNA analysis. And I guess it shows you, so for this case, we actually used all the techniques that we had on hand. So we even used BioNano, we couldn't figure it out. We used short read whole genome sequencing, and we also couldn't figure it out. So this, I think, shows you, I think, also the strength of combinations like using RNA and long read DNA. As one more example, I guess this is, I think, nice playing around since we have the data anyway. So if you look at the SVA insertion, what you see is that the promoter of the SVA is actually also hypomethylated, which is to be expected. And if you look a little bit closer to the SVA, the ALU elements, then you can see it's also methylated. Okay, so one more. So as I mentioned, this is diagnostically confirmed, and I guess this was used for a family member who was expecting a baby. So then based on these diagnostic confirmation of this test, they can use it as a prenatal test basically, or to figure out if somebody has a chance, risk of getting this disorder. So one more example. This was a family with 3 unexplained cases of a disorder called Alagille syndrome. I don't think it really matters what the phenotype of the disorder is. But the point is that these are typical diagnostic questions we get. For example, in a SNP array, they find some weird abnormality. They find there's one causative gene for this disorder, and they find in the SNP array, they find a deletion next to the gene. So it's annoying, right? So you, just want to, it's not a confirmation, it's not a diagnosis, but it's, so what's going on there? So we performed RNA sequencing, and we had a really weird finding. So the gene next to, so what you see here is the Z-scores of the transcript plotted over the x-axis basically, and the gene JAG1 is the gene related to this disorder, and the gene next to it, part of the gene is up and part is down. So that was surprising. So we looked a little bit closer, and it turns out there's a fusion transcript. So there's more going on than a deletion next to the gene. So based on a lot of experiments, we figured out there must be something going on like this, maybe a duplication, maybe also an insertion. So there's probably more going on than just based on the SNP array. And then we went to Nanopore, and this is actually what I think it really nicely shows you what's going on. So this is the Nanopore data, and what you can see here is the gene next to JAG1 and the gene, and JAG1. And what you see is that there's a big deleted. This part here is deleted, so we already knew that. But there's also a part of the gene next to it that's actually inserted in an inverted way within the JAG1 gene. And I think all the breakpoints are line repeats, so there's some kind of messed up event going on here. So we think that's not just a deletion, there's also a proximal insertion of an inverted part of the deleted region. So these are, I guess, typical things that are, even with this Nanopore data, it took really a long time to figure out what was going on here. So with that, I would like to acknowledge the people involved, mostly the people on the left. It's our innovation team, the people involved in implementing nanopore sequencing in our lab.